Hello, welcome to the showcase for the tidyCDISC application, which is an interactive R platform to analyze clinical trial data. It is currently open source on GitHub. My name is Aaron Clark. I'm the primary developer and maintainer of the package. So what is it? It's a pharma-specific Shiny app that we've packagized so that users can use it within their organization to interactively explore tables, figures, and patient profiles using clinical data, often called ADAM datasets. There are three major features. The drag and drop table generator, there's a population explorer that basically creates visualizations, and a patient profile viewer, which essentially allows us to zoom in on one patient's journey through their clinical trial. So how do we do this? We're going to rely heavily on the CDISC standards, which are basically a structure that we all as pharmaceutical companies agree to follow so that our data looks a consistent way from company to company. Which means we can leverage relevant analyses to prepare visuals and figures using our favorite tidyverse package, hence the name tidyCDISC. If you check out our GitHub, you can see that the languages that we leverage for this app include 70% R, 26% CSS, and a little under 4% JavaScript. All right, so if you're a user in the tidyCDISC application, you're going to notice we're asking you to upload your ADAM datasets. So your data could look like this. Maybe you upload 3 datasets. An ADSL is sort of your high-level dataset with 1 row per patient. Then maybe you have a vital signs dataset and a lab dataset with different parameters. What we're going to do inside the application is sort of rearrange that into an internal data structure that we can use to quickly and easily prepare tables and figures. So why do we need tidyCDISC? Well, besides the fact that it creates additional value around those CDISC standards, it's also going to eliminate a ton of unnecessary communication by providing self-service analytics. And thus, it's going to save a radical amount of time. Consider this use case. Let's say we have a fictitious clinical trial team with a clinical head named Ika, a statistician named Will, and a programmer named Monica. So generally how this works is Ika is going to meet with Will and create the statistical models to be used in the study. Will is going to ask Monica, the programmer, to create the tables and figures using these specs. Monica is going to deliver on those requests, and then Will is going to say, hey, you know what, I actually need a few more based off of the initial results that Monica provided. So Monica will provide those new tables and figures and send them back to Will. And then Will's going to ask for a few revisions on those tables and figures. You can see that this can go around and around for a while, but eventually Will is going to talk with Ika again, showing her the outputs that they prepared. This is where tidyCDISC sort of enters the field, essentially allowing each user, Ika, Will, or Monica, to explore the data easily on their own. So this is useful for Ika because she has the most domain experience, being the clinical head. Will, the statistician, of course, can use tidyCDISC to make tables and figures almost instantly, and thus he doesn't need to ask Monica to program the tables more than one time. Monica can use it for QC purposes. So before she begins to program the tables and figures in a validated software, she can use tidyCDISC to preview them or compare them against what she's doing. Okay, all in all, we've had users report time savings upwards of 95% by using tidyCDISC while performing their general programming duties. Now I'm going to take you on a walkthrough of the application. If you're interested in taking a spin yourself, it's currently available at bit.ly/tidyCDISC_app. And again, we have rich sources of user guides that are currently hosted on our GitHub. So if you'd like to take a look, there's lots of things in there, including a purpose statement, links to a getting started vignette for first-time users, and tons of articles for those who want to dive deeper into the application. I'd be remiss if I didn't acknowledge the team that's worked on this project before I start my demo. So there's myself, Aaron, also Bob Engel, Maya Gaines, Marley Gotti, Robert Krashik, and Nate Mockler. I'd also like to solicit your help. If you're interested in tackling an open issue, we would certainly be open to reviewing a PR, or if you're interested in pioneering this application at your pharmaceutical organization, we'd certainly love to join hands with you and work on it together. If you have any questions, feel free to reach out to me via email or LinkedIn. I'd be happy to chat with you. Without any further ado, here's the demo of tidyCDISC. I'm going to go ahead and open the demo version of the app that's hosted on Shiny Apps IO so that you can follow along with us. As you can see, the data tab is the landing page for the app. The demo version of the app actually uploads the CDISC pilot data by default and disables this file browse widget. So if you want to test your own data in this application, what you have to do is go back to GitHub, scroll all the way to the bottom, and follow the easy installation instructions for tidyCDISC. All you need to do is run remotes, install GitHub, Biogen Inc./tidyCDISC. Okay, so you can see we have a collection of 5 files from the CDISC pilot data. And this data previewer just allows the user to have a perfunctory overview of the data before navigating to an analysis module. So let's jump over to our first module, which is the table generator, which I would say is probably the bread and butter of this application. Now, the table generator leverages a pretty unique drag and drop interface that allows the user to interact with all of the data frames that have been uploaded on the data pane and marry them or pair them with a statistical block. So for example, if we were to check out the ADSL, we could scroll through and look at all the variables in the ADSL. Maybe we want to pull out age, and we're going to drop that into the variables drop zone. And then we will pair that with a mean block. And doing so is going to produce typical descriptive statistics we're used to generating for clinical trials. And however, if we want to generate maybe some frequencies for a categorical variable, it's easy to do that by dragging in the categorical variable like age group 1 and pairing that with a frequency block. Notice how that any additional layers get appended to the end of the table, but you are allowed to move those around to your heart's content. You can even get rid of certain rows just by clicking these X's. Now, let's say that we're interested in analyzing some data from a BDS data set, such as an ADVS. Here you can see not all the variables are listed, but instead all the param-CD codes are listed. And when you hover over them, you can see the SAS labels in the underlying data. So let's say we'll grab diastolic blood pressure, and we'll also grab a mean statistic block. And notice that we don't get an analysis until we actually select the week at which we'd like to analyze diastolic blood pressure. So here we'll select baseline, and that will update our table output. So unfortunately, we won't be able to go through all the statistical blocks that we've generated for the table generator. But if you just hover over them on your own time, you'll get a quick description of exactly what they do. Or if you want to consult the documentation on our GitHub site, it's very, very thorough. So now I want to turn our attention to ways in which we can slice and dice the data. So for example, if you wanted to display these same metrics by males and females, you could do that by grouping on sex, or you could select maybe one of the treatment variables, and you can see the placebo, low dose, and high dose groups are selected, and they all have their own column with their respective ends, which is very, very handy. Another way to slice and dice the data is by filtering. So here, if I select the ADSL data set and a variable from that ADSL, I can now filter all of the data to patients who are greater than or equal to 80 years old, or in this case, maybe we want to say 81, so we can get this one subgroup. So those are different ways to slice and dice the data. We have these things called standard analyses tables, which actually, if you click one of these, it's going to pre-populate your table with standards for demography. So in this case, we lost all the old blocks and they were replaced with new ones. So here you can see we have summary counts for age group, age, sex, ethnicity, race, and height. Similarly, we have oodles of adverse events data sets, such as overall summary. We have system organ class by preferred term, and so on and so forth. So let's say that you're really content now with whatever table you've either built manually or a standard analysis table, and you're ready to share it or store it for later. So here I'll just say, this is a pilot data table, and it's for demography, and we can download that as CSV, or we can download it as HTML to use for later, or maybe share with one of our coworkers. You can see that it looks exactly how it was produced in the application. We take this one step further by providing the R script that's needed to produce this code in an R session. So outside of the app, on your own time, perhaps after the data gets refreshed or something like that. So here's the script that I just downloaded from our browser. And you can see that it's going to download any packages you need, including the tidyCDISC package from GitHub. It's going to load those packages, and then it's going to help you load your data sources. And I'm just going to click source, and that'll run through all the code. You can see all the code's running here, the bottom right-hand corner. And that produces our final table. So pilot data demography. And it's basically the demography table grouped by treatment. That's exactly what we were hoping for. All right, so I'll minimize that. And that's basically the end of the tour of Table Generator. One thing that I wanted to point out is that everything is very modularized within this application. So if you come up with an idea for a new statistical block or a new standard analysis table that you want to add, go ahead and open a PR. We would love to talk about that, or file an issue. If you see the top right-hand corner, there's a little GitHub icon. If you open that, it'll bring you right to a new issue on Biogen's GitHub page. So there's that. And lastly, I just want to note that we have these cool little question marks everywhere, all over the application. And whenever you're feeling stuck or you don't know how to use the application, you can click on that, and it'll actually bring you through a guide about what each element of the user interface does. And it'll literally bring you around the app, showing you thing by thing. And that exists on all tabs: the data tab, the table generator, and population and individual explorer too. All right, so our next module, I'm going to go ahead and hop over to the population explorer, which features some of the same filtering technology that we use with table generator. But essentially, this module is used to visually inspect the data for the entire population. And we have a number of plots here that you could scroll through. I'm only going to have time to go over a few of them today. So because we do have a time to event file uploaded, we can check out the Kaplan-Meier curve. If you don't have this data uploaded, this chart type will not be available. You can add 95% confidence bands on there. The plot is very, very interactive. So if you wanted to look between certain time points and zoom in on those, you could certainly do that. You can zoom out, you can download a PNG of the image. And what I really like is you can separate it by different groups. For example, if you wanted to look at, I'll get rid of the confidence bands. If you wanted to look at males versus females, or if you wanted to look at our treatment groups like we did before, then you can do that as well. All right, I just wanted to show you briefly, really popular plot is to look at a mean of certain parameters over time, so by visit. So I'll look through this list and just choose one of these parameters. And it's also going to prompt me for a metric, either eval or change. I'll leave it on eval. And then I will select one of our time variables. So this is going to plot mean calcium by visit based off of what I've selected here on the left. You have the option to overlay vertical or horizontal lines. If you want to compare to a certain threshold, you can do that. I'll just show you what that looks like. Yep, there's the baseline. If you want to move the line to week 2, you could do that. What's really cool is you can display each of these as a 95% confidence interval. And as you hover over each of these lines, you'll see all the upper bound, the lower bound, and the center. You can also label the points if that's something that you desire. In addition to that, I find that slicing and dicing the data is very helpful. So just like as before, you can separate it by males and females. And then if you wanted to separate your plots, you can actually do that by treatment. And that allows you to get quite a bit of depth from this visual, because you can look at across all groups, how are my males and females performing, or sorry, within each group. All this data is available for download to Excel directly beneath the graphic. And as always, you can download a PNG for later viewing or for sharing. All right, last but not least is Individual Explorer, which allows us to sort of hone in on a single patient's narrative through the study. If you don't know the patient use subject ID, that's okay. You can actually filter to different groups. So just like in the last couple modules, you could say, hey, I want to look for patients who are over the age of 80. Our patients, and you can see here, there's 254 patients. If I go to 81, now there's only 71 patients left in the trial. And that means that this list only has 71 use subject IDs instead of 250. I'm just going to grab the first patient, and you're going to see off the bat we have some demographic info. It's a white female, age 63, in the placebo group. There's two things I want to show you. There's an events tab and a visits tab. So events basically is just going to grab all the important dates from any file that you've uploaded. And from there, you're going to be able to see everything on one interactive timeline. So you can see here the patient had onset of disease in 2010, and they were enrolled and had first exposure to treatment in 2014. Each event sort of has its own swim lane. So we have milestones, lab results, and adverse events have their own swim lanes. You can zoom in here to see all the chem labs when they happened, and you can even check out all the adverse events. All the data, again, available below the chart. You can download it to Excel. But what's not here is probably comparing these important dates versus some important metrics that we maybe got from those chemistry labs. You know, we're drawing these chemistry labs. It'd be nice to actually see what's happening at those visits. So here's an example. Let's say we go back to, this is sodium, but let's say we go back to calcium. And we select one of our visit variables. We'll just leave it on ADY. We can plot a horizontal line to see how each week compares to the baseline week, and we can overlay our adverse events here. So here you can say they had a few early on in the trial. From there, you can download all of this information directly to Excel. And as I mentioned before, there's a lot of parameters in here. So if you just want to get a report for this patient that has all the parameters listed, you can do that very easily, downloading to PDF or HTML. So I'll go ahead and just download the HTML. Give that a few seconds, and it should pull up for us here in the browser. And this is helpful because you can take it and look at it later or share it with one of your coworkers. And it's easy to just sort of breeze through, and you don't have to manually click through each chart. Okay, so here's a summary, 18 parameters for this patient, and here's all the graphics. And just like in the app, since this is HTML, it's all very interactive, and you can still zoom and download PNGs and all that jazz. What's nice is you have a nice table of contents to be able to move around quickly. All right, so that concludes my demo. I'd be happy to field any questions, but if you'd like more info, please check out our user guides that are on our Biogen GitHub page. If you scroll to the top and click on this link, that'll actually bring you to our very, very exhaustive, in-depth user guide, where there's a Get Started guide, and there's also articles on each tab that does a deep dive into each tab of the application. So as always, we'd love to hear from you. Please submit an issue or a pull request, and thank you so much for listening.